My name is Gage. I'm a faculty member at the Salk Institute and the Adler Professor in the Laboratory of Genetics. So mental health diseases like schizophrenia and bipolar are really unmet needs in the population today. Roughly 8 million people in the United States are suffering from these diseases, and there are very few treatments for these patients currently. Psychiatric diseases have been hard to determine so far the cellular and molecular mechanisms that cause the disease. And by having these new tools to examine the neurons from a patient in a dish, we are hopeful, and there's evidence for finding some of these molecular markers for the disease that have been elusive in the past. We can now take cell biopsies from individual patients like yourself and grow the cells in culture, and these are just normal cells from the body. But by using a chemical reaction, we can convert these cells into very primitive cells called embryonic stem cells or embryonic-like cells, and those cells then can be converted into neurons. Now we can measure neurons from any individual, and they have within those cells the genetic makeup of the disease. So we can now examine more directly human disease, neuronal disease in a dish. Researchers, including our group and others, have now used this technology to begin to search for molecular mechanisms that underlie psychiatric diseases like bipolar and schizophrenia. Now, this technology is relatively new, and certainly its application to psychiatric diseases is new. So the National Institute of Mental Health has initiated a program, and they're the ones that are actually supporting this grant, to bring together universities from around the states, and in our case we have the University of Michigan, Johns Hopkins University, along with Salk Institute, to working together using protocols and assay systems that we've developed in our lab to find differences between patients that are affected and unaffected with the disease, and develop these in a way that are most reliable and applicable. After we have developed these techniques in a way that allow us to probe deeper into the diseases and identify specific targets that might be the basis of the problem, we want to be able to help develop therapies for these diseases. So as part of the NIMH grant, consortium grant that we're developing here, we also have involved industrial partners from pharmaceutical companies. And now it's academics working together with pharmaceutical industry to see if the assay systems and the discoveries that we make in the lab can be effectively translated into systems that they can use for drug development. This collaborative arrangement between multiple universities along with industry will be led by myself and Hong-Jun Song at Johns Hopkins University. I might add that Hong-Jun Song 20 years ago was a postdoctoral fellow here in my lab at the Salk Institute and is now a full professor at Johns Hopkins University. This consortium grant is funded at $15.4 million over a 5-year period of time. The members of the consortium include the Sanford Burnham Prebys Institute here on the Mesa, along with Johns Hopkins University in Baltimore, the University of Michigan, as well as Johnson & Johnson, in particular their campus here in La Jolla, along with Cellular Dynamics, based in Wisconsin. We've been working and developing techniques in this area for some time, and we're very excited about being able to continue with our work, but also collaborate with such outstanding colleagues.